All right, we're going to begin our last unit of the semester with Unit 3, Lecture 1, the Human Microbiome. And this happens to be one of my favorite subjects. So what we're getting into here has to do with how the body is actually known today to be a collection of different organisms working together rather than just the human body working by itself. So let's take a look at the first slide here. Only very recently, in the grand scheme of things, has this distinction been made. So humans have known for a while that we are made up of many, many cells. Today we know that to be roughly 30 to 100 trillion cells, depending on who you ask. And we have also known for quite some time that there are microbes that live within our body. This has been discovered pretty much since the beginning of the microscope itself. Anton van Leeuwenhoek was one of the first ones to really show that the body had microbes in it by taking scrapings of his teeth, for example. And he was able to show that there were little, what he called animalcules floating around in these teeth scrapings. And that was in the 1600s. And since then, more and more research has been done to show that, yes, in fact, there's microbes kind of all over us. We could swab our body and grow microbes. We could look in our gut and find microbes. And so this has been known for well over a couple hundred years at this point. Now, starting in the late 1800s, early 1900s, a lot of that research began to focus on harmful microbes, pathogens. And it was shown that germs could cause disease. And this was known as the germ theory of disease put forward by Louis Pasteur. And over time, the emphasis really grew around harmful pathogenic forms of microbes, bacteria and viruses primarily. And so throughout that time frame, it became more and more clear that yes, you have microbes and these microbes can be bad, and ultimately we have to focus on these bad microbes. And there was good reason for that, because it was true that microbes could cause disease. However, as time has gone on, and really only until maybe the 1980s, starting in the 1990s, but really advancing significantly starting in the early 2000s, has it been shown that that relationship is actually much more complex. And it's not really just a matter of bad bacteria infiltrating the body, therefore causing disease. While that is true, it actually turns out that we have microbes that have to form a balance within the body. And that balance is just as important to maintain in order to maintain good health as it is to avoid the presence of bad bacteria. So in other words, you have good bacteria that have to be maintained, and that is a key factor in your health. And that is just as important as it is to avoid the presence of bad bacteria infiltrating your body through infection. So, in fact, this has been so overwhelmingly clear at this point that a new term has been coined to describe this, and that is called the holobiont. And what that sort of is almost a symbolic gesture to this concept, which is that science now recognizes that the human body is really a construct of both its own cells and the microbial cells that live within it. So the term holobiont basically means the combination of the two, kind of indicating that you can't actually just look at the body by itself. You have to actually understand it in the presence of microbes that make up the whole, the holobiont. So we have microbes on the skin, in the mouth, in the digestive tract, and in the urogenital tract, amongst others. And so that idea has now really taken front stage in the study of the body, microbiology, and really health and disease in general. Now, these microbes are very, very complex, and so there are many different types, but we call these symbionts or symbiotic microbes. Now, what seems to be the case is that bacteria are by far the most common, but also really the most well studied. So as of today, it seems clear that when you look at all these microbes, bacteria are the most important. But there are also a combination of archaea, fungi, viruses, even mites that could be found almost exclusively in the eyebrows, for example, and even a range of different protozoa. But most of this research has largely been focused on bacteria. So the term that you kind of then use to describe the microbes, or I should say terms, varies quite a bit. And just a few of those include things like the microbiome, the natural biota, the microbiota, or even the human microbiome. And they all pretty much mean the same thing, but there are different ways of describing them. So, as I mentioned, most of the research to this date has focused on bacteria, and that is because when you try to analyze what's there, bacteria seem to outnumber all the other microbes by quite a bit. Now, there is a caveat to that. New research has shown that viruses that infect bacteria may in fact actually outnumber the bacteria themselves. So you could argue that the viruses are actually the most common, and there's a nuanced argument to that. But ultimately, those viruses are typically the ones that infect the bacteria, and they tend to outnumber the bacteria. So putting that aside, generally speaking, when you're talking about the microbiome, you're largely focused on the bacteria. Those seem to be the most prevalent form as of what research has shown today. Now, going back, I said, a couple hundred years, no one really seemed to understand this or appreciate it, and so that has changed relatively quickly, really within the last 20 years, or really even less, depending on exactly who you ask. So ultimately, scientists will often agree that within the last 20 years or so, we've kind of entered this new era of microbiology, kind of going from the previous mindset of microbes cause disease and that's their main importance, to now kind of microbes are actually part of the body, and you almost have to maintain a good balance, almost like you would maintain your lawn. You can't just let your yard and your lawn be unattended. You have to actually keep it maintained and nourished and all of that. And so that's kind of a way to think about the microbiome, is like a flora that you have to maintain and not ignore, and not only pay attention to when there's a problem. So a couple of things have changed this. The first one, you could argue, is technology. One of the reasons why it was not known that these microbes existed, or at least in the way they exist, is because of a lack of technology. And the biggest improvements which have come in the last 2 decades are DNA sequencing techniques. So today, scientists can really analyze the world through genetic sequencing. And so this allows scientists to take DNA from the body and analyze it, not only just our own DNA, but the microbial DNA itself. And this then leads to what's called metagenomics, which is just a more complex way of analyzing large amounts of data. So you can take these bacterial genome sequences, analyze them through computers, and as a result come up with large amounts of data about what's actually going on inside the body. And that has revolutionized the field, because prior to doing this, you weren't able to really tell what was there. Along with that have come significant reductions in cost. The ability to sequence these genomes has gone down in cost, and you can see some of the numbers here. One of the first big projects where they did this was called the Human Genome Project, and the cost was tens of millions, up to $1 billion. And within just half a decade, that cost had dropped significantly. And the last estimate I have here from 2016 puts that around $1,000 to $1,500. So going within about a decade and a half, from millions to thousands of dollars, and that number drops even more and more each year by a little bit. So the technology itself and the reduction of cost have been a big aspect of our ability to even have the potential to analyze this data. But in addition to these technological advances and reduction in cost, another big issue is awareness. Prior to really the early 2000s, no one gave much attention to the collection of microbes that were in the body outside of those that cause disease. So starting in the early 2000s, microbiologists and scientists in general started to actually pay attention to the fact that, hey, you actually have all these microbes there, and it's starting to look like they have an impact on health and disease in general, and it's not just necessarily the presence of germs that cause disease. And that was a departure from the previous several centuries, where the emphasis had largely been placed on the sick and the dying. And for good reason. You know, germs, we well know, cause death and disease, and so it made sense to focus on that. But today, what we now are starting to recognize is that you also have to pay attention to why people are healthy, and not just the fact that they lack the presence of a germ. Germs definitely do cause disease, but the ability to maintain health is not just a factor of lacking the presence of a germ. Okay, and so while we made lots and lots of progress during what some people call the golden age of microbiology, we are now sort of entering into this new era where the emphasis is now more being placed on the normal status of the collection of microbes and their contribution to overall health and body fitness. And so that's kind of where the new era has gone. Why are you healthy, and how does that relate to your microbiome? And what might go wrong with the microbiome that makes you become unhealthy? And the emphasis is not just on the introduction of a germ as part of that equation. So when you look back at this golden era, lots of important discoveries were made, but they did not realize at that time that the vast majority of microbes on the body could not be cultured. And this was a big reason why they didn't necessarily understand to pay attention to these other microbes, because they actually didn't know they were there. It turns out that only about 1% of microbes can actually be cultured on some kind of growth media, like a petri dish. And there's lots of different forms of media, and you can use different forms to culture more or less microbes. But the estimate today is that no matter what kind of petri dish culture form you use, you can probably only culture about 1% of everything that's out there. And that's where the genome sequencing I mentioned earlier has been so important, because you can analyze a microbe by looking at its genetics without ever having grown it. And so you can reveal the presence of microbes even when you can't grow them on a petri dish, which has kind of opened the floodgates to this other 99% that were previously unestablished, and no one necessarily realized that they were even there. So that has then ushered in this concept that the quote-unquote normal biota might cause disease. In other words, it's not necessarily the introduction of a germ. It is the fact that the bacteria that have always been in your body are starting to change or become altered or doing something that leads to this sort of subtle, gradual, chronic type of disease process that is actually a manifestation of the normal biota gone wrong, so to speak. And so this is sort of the idea that these microbes can cause subtle abnormal conditions, as opposed to an abrupt infection that comes about suddenly due to some serious pathogen. So some of the disorders that are being studied are things like depression, anxiety, obesity, and others that I didn't list here would include things like autoimmunity and cancer and many other types of specific organ diseases, liver failure, colon disruption, and things of that nature. And so that's all kind of going together. We didn't know they were there. Then we know now we know they're there. Now we can pay attention to the contribution, and now it looks like they're causing subtle, sort of gradually increasing chronic disease over time. So with that knowledge being now known, scientists are really studying what is it about the combination of microbes in the body that contributes to the overall health of the human individual as a construct of not only its own cells, but its microbial community as well. Now, the problem here is that this is a very, very, very complex relationship in that no two people have the same microbiome. So it makes it very difficult to analyze what exactly is happening. So this will most likely be a multi-decadal process, could be much, much longer, to really sort of whittle down to what is exactly happening. And in all likelihood, you won't find conclusive answers that apply to everyone. You'll have to sort of tailor the diagnosis to the individual. But that's largely speculative, because at this point, mostly just broad generalizations are being made. So in that sense, the study is in its sort of quote-unquote infantile stages. And to give you an example of how some of these concepts are changing, there's a statistic that's often stated even today, and that was in textbooks for many, many years, which was that the microbes in the body outnumber our own cells 10 to 1. That statistic is still cited. I see it frequently today, and was a common stat given in textbooks. And the idea was that for every one cell in the human body, there are 10 times as many cells of microbes. And what that actually came from was a particular study done in the early 80s that was then picked up and published and then put into textbooks and then cited from the original source over and over and over again. So it looked like you were seeing many, many sources all confirming the same thing, but in reality what you were seeing was one source being pushed by many, many different entities. And that statistic has now been shown to not only be wrong, but way off. And so today it's now recognized that while you do have lots and lots of microbial cells, that number or that ratio is actually closer to 1 to 1. And it may be slightly higher, 1.2, 1.3 to 1, but it's much closer to a 1 to 1 than it is a 10 to 1. And so it's just an example of kind of more change, correcting some of the previous misstated stats and kind of updating to a more closer approximation of reality. So it's now recognized that if you have, let's say, 50 trillion cells, then you'll have just slightly more than 50 trillion microbes in the body. And that can vary from person to person. It's not an exact stat, but it's closer to 1 to 1. So just another example of how things are changing here. Now, one of the biggest sort of landmark studies that kind of put most of this into motion was called the Human Microbiome Project. And it was done in the, sort of mid to late 2000s and had finished up roughly a decade later, and now you have many sort of offshoots of that study happening. So this particular project is not the end-all be-all, but it kind of started a lot of this. And so what they were doing was the genome sequencing I mentioned earlier, and they started off with the goal of just collecting genetic sequences, trying to figure out what was there, focusing on these unculturable microbes, recognizing that only a small fraction, about 1%, can actually be grown, and then ultimately kind of expanding that into the future, looking at what role these microbes play in health and disease. Now, for the most part, the human microbiome did do a very good job of accomplishing those first 2 points of sequencing and focusing on the unculturable, but the ability to connect that to health and disease turns out to be a lot more complicated. That's going to be an ongoing process that maybe never ends, but certainly will take many, many more decades. But that's really where a lot of this started. Now, one of the things, a handful of things that they showed here was that the human body contains roughly 20,000 protein encoding genes. This number, 22,000, has been revised down a little bit, and it's not an exact number, but we can say that we have a roughly 20,000 protein coding genes in the human body. In other words, the human genome produces about 20,000 different proteins. And we talked about that a little bit back in the genetics, where we talked about structural genes or protein coding genes. So if you kind of take that as a general estimate of how many different proteins you can produce, and we know that proteins do work in the body and make up structures and enzymes and things like that, it kind of gives you an idea of how much the body itself contributes to its overall metabolic protein status. Part of what the human microbiome started to show was that if you look at the microbes in our body who also produce proteins within our own ecosystem of the body, their contribution could be roughly 8 million protein encoding genes. And that's a rough estimate. You're going to see different numbers from that. But much, much more is the main emphasis there. And according to this estimate, that means that the microbes within your body could have the potential to produce 300 times as many proteins as your own body. And what that means then, if we look back at the concept of the holobiont, that not only do both the microbes and our body contribute to this complex of proteins, but the microbes seem to produce many, many, many more proteins than our own body can produce itself. Not all of those are going to turn out to be significant, but even if a fraction of those do, then we're looking at many, many, many more times contribution from the microbiome to this overall protein makeup in the body than our own cells do. And so that one stat there, if you really understand that, kind of helps you understand why these microbes are so important. If proteins do work in the body, and the total collection of proteins are both microbial and human, then you can kind of start to see why these are so important. So that was probably the biggest fundamental breakthrough, that our body needs these microbial proteins in some way to be healthy. Another big contribution was areas in the body that were thought to be sterile were then shown to actually contain a microbiome or microbiota of some kind. And that was largely because they couldn't culture anything there. If you tried to culture the lungs, for example, you generally wouldn't culture anything. So they said, well, it must be sterile. After doing genome sequencing, they found, no, there's actually microbes there. It was also found, interestingly, that you would find dangerous pathogens in virtually all the healthy people that they studied. And that goes back to this idea that it's not necessarily just the presence or absence of germs that make you sick, because these healthy people had quote unquote germs in their body, and yet they didn't have disease. So why weren't they sick if they had these germs that were present? And that has been also revealing that there's more to it. You know, it's not just the presence or absence of a germ. It is a combination of other factors that contribute to whether that germ actually makes you sick or not. They found viruses in healthy individuals. A lot of these are bacterial, but there are other viruses that are non-bacterial as well. Also, again, it's not just that you get a virus that makes you sick, it's that you have viruses that are part of the body that are there naturally. And then the biggest influence has been your bacteria in the gut. That seems to be where most of the emphasis today lies. Bacteria are found throughout the body, the skin, the mouth, the reproductive organs, but most of them are actually in the gut and in the large intestine in particular. And that seems to be where they have the biggest contribution. So a lot of the emphasis is on the gut bacteria and gut health in relation to gut bacteria. So they found connections between obesity and specific autoimmune disorders like Crohn's disease, for example. When it comes to obesity, they found that the types of bacteria in your gut contribute to your overall body weight. So if you have certain strains, you're more likely to be skinny, and if you have other strains, you're more likely to be obese. And so that was a big contribution just by itself. And then other research has sort of led to the idea that gut bacteria can affect everything from heart disease, asthma, autism, diabetes, and psychological mood disorders of many kinds. So that is really where a lot of the emphasis today is placed, is on the specific types of interactions with the gut and the gut bacteria. So here in a minute, we're going to talk about where you kind of start off acquiring the bacteria and that in the microbiome, which is when you're first born. But as an adult, you have sort of an established microbiome by the time you're even a few years old, and it does change over time. But ultimately, every day you come in contact with new microbes, whether that's through food or air or exposure through contact and things of that nature. And so you're constantly being sort of inoculated, if you will, through microbes. And because of our immune system, most of these microbes are removed or destroyed long before they ever get a chance to establish, which means to kind of take up permanent residence. Those that actually do establish will ultimately be removed over a longer duration of time by the same defense system in more specific ways, the immune system, essentially. And we talk about that more in the coming chapters, how exactly that works. But you get introduced, and the body tends to remove those relatively efficiently. However, a small percentage of those that are not removed will actually take up some form of permanent residence in whatever body location, gut, skin, oral, reproductive tract, and then tend to reproduce and colonize in some kind of permanent or semi-permanent fashion. And over time, then, you start to produce this sort of ongoing symbiotic relationship with those microbes. And these ultimately become what we call the mutualistic or commensal microbes in the body. The term commensal is actually a common way to describe it, but mutualistic means that they'll be beneficial to you, and commensal just sort of means that they'll reside there and may or may not have any benefit to your body. So ultimately, we get in exposure, most of those are removed, a small percentage of those established to create these relationships, mutualistic or commensal. Now, when you go to look at the body, what you'll find is that any given body location will have microbes with few exceptions. Like the central nervous system is an example where it doesn't appear that you have normal microbes within the brain and the brain stem and places like that. But outside of that, you're going to find microbes in almost every area. But they vary from person to person and even from location to location. So no two people will have the same exact collection of microbes. And even within one person, you will have different collections in different parts of your body. And this could actually even be on similar body locations. So like your right hand will likely have a very different collection of microbes than your left hand, depending on the dominance of your hand and just what you're doing, which kind of makes sense. If you're right-handed, you're going to touch more things with your right hand than you will with your left hand. And so it actually turns out that just within one person, location to location, the microbiome can vary significantly. But what is actually more important is not necessarily the exact species and the exact name of the species and all of that, because that is what ultimately differs. But what is actually fairly consistent is the protein profile. So it turns out that microbes, for example, will colonize the skin, not necessarily because of what you call the microbe and the name that you give it, but because of the kind of protein profiles that those microbes produce or the kind of enzymes that they produce. So the skin is a specific environment. It's somewhat acidic, it's somewhat salty, it's kind of dry, it's not as moist in that particular area, especially on the outer layer. And so in order for a microbe to survive there, it has to have certain adaptations. It needs to be able to make certain proteins and enzymes, and it has to have certain features that allow it to survive in that environment. Kind of like the difference between a fish living in the water and a lizard living in a dry, arid desert landscape. They have different features that allow them to survive in one or the other. Generally speaking, that's kind of the same for bacteria. So while you may find differences between one person and the next, you're going to find similar types of microbes based upon the kind of protein profiles that they produce. And so that is actually shown to be fairly consistent. So the species and the diversity of species varies, but the kinds of microbes based on their protein files tends to be fairly consistent across most people that were analyzed. So that's a kind of an interesting thing, which makes sense. I mean, you may go to a desert and find different lizards living in different deserts, but you're not going to find fish. You're going to find lizards because fish can't live in deserts, obviously. So that's sort of the similar concept here. You're going to find different but similar types of microbes depending on where you look. Now, where these microbes start to get more complex is especially in the gut in particular, where a lot of this interaction takes place. They can provide both benefits and harm, largely in reference to what's referred to as microbial antagonism. So this is kind of a technical term that refers to the inhibitory effect that the sort of normal microbes have on other quote-unquote invading microbes. And in this sense, you might almost think of it like good bacteria act as bodyguards for the gut. And this could apply other places as well. But they kind of occupy the gut, they don't cause harm, therefore they're not harming you, but even more, they prevent microbes that might cause harm from coming in and establishing. And so in this sense, they're antagonistic to outsiders who might actually cause harm to you. So a lot of this is done just by blocking body sites. There's only so much surface area that a microbe can occupy. And so if the surface area is occupied by good bacteria, then there's no room for bad bacteria to come in. And as a result, they kind of naturally, antagonistically prevent establishment or colonization. But a little bit more complex, part of what goes along with that is they can release chemicals that are also harmful to invaders, and so they kind of create an environment that's hostile to outsiders. And so the combination of those two create what we call microbial antagonism. And this is sort of a visual concept of what that might look like. This is a petri dish with one kind of bacteria here preventing the colonization of what is actually a fungus here. And you can see that this little ring between the fungus and the bacteria is clearly a separation point between the two, and there are actually antagonistic chemicals being produced by the bacteria that make it impossible for this fungi to grow in this exact area. And so you can kind of see that on a petri dish. There's a much more complex interaction in the gut, but that same kind of effect is taking place in a more nuanced way inside the body. So the normal biota are now recognized to kind of aid the body by at least preventing harmful microbes from coming in and taking up residency. Now, the normal biota is a balance, and this is where it starts to get a little more nuanced. So it's not necessarily just having the right species and the right types. It is also about having the right numbers. You don't necessarily want too much or too little. You want to have everything kind of finely tuned, so to speak. So the balance turns out to be more important than just having the right kinds. So you don't want overgrowth or undergrowth of any one particular species. Now that's one of the areas of research that's going to take more time to figure out what that balance is exactly supposed to be and how that varies from person to person. But that's just sort of an important note here, that it's not just one type or the other, but the right ratio and things of that nature. And one of the problems that has already been established now for a while is that taking antibiotics is almost like a nuclear bomb to that balance in the gut. The body takes time to establish these complex relationships, and antibiotics, which can be useful, are harmful in the sense that they tend to broadly kill microbes indiscriminately. So if you have an infection and you take antibiotics and you swallow those pills, the drug goes down into your intestines, into the gut where most of these microbes live, and they tend to indiscriminately kill most of the bacteria there, and almost like burning down an entire forest ecosystem. And the forest will eventually rebuild itself and regrow, but it will not necessarily regrow itself into the exact same balance of organisms that it had prior to that devastation. And so the same thing is largely true of antibiotics. You have this sort of long ongoing development of these complex relationships, and then almost overnight you can just wipe that entire relationship out, and now the body has to rebuild itself. And while it can do that, it can also go wrong and not be able to reestablish it back to where it was before. One common example of that is with C. diff, or Clostridium difficile, which I'll mention here in a second. But this is a figure kind of showing in a way that you have the normal gut ecosystem here, healthy, you're eating certain foods, you get these certain types of complex balances. The immune system, which is what's shown here, is kind of responding to those in what we would call a healthy kind of way. You take antibiotics, that wipes out a lot of species, significantly reduces the microbiome diversity, and now all of a sudden the immune system isn't responding as well. The entire ecosystem has changed, and as a result, disease can result from that in very complex ways, and it's hidden from view, so it's very difficult to establish what's happening there. But that's kind of a generalization. And when you see that with C. diff, here's kind of an animated illustration of that. You see that you kind of have normal gut lining. These are like going back here, these are like the cells of your gut lining. And what this animation is trying to show, or illustration, is that over time you introduce these antibiotics, they sort of allow C. diff to flourish, and then as they flourish, they start to release harmful toxins that affect the normal gut lining, which becomes diseased and then over time causes its own set of problems. And one of the reasons why this happens with C. diff in particular is that it's an endospore forming bacteria. And it isn't necessarily killed by the antibiotics in the same way that other bacteria are. So the C. diff can be somewhat immune to the effects of the antibiotics. The antibiotics wipe out the normal bacteria and leave lots of room. There's now open surface area that was otherwise blocked. C. diff can now start to grow. It becomes heavily imbalanced, and as a result, you get this disease in the presence of an abnormal number of harmful cells. Going back to what I said earlier, you can find C. diff in healthy people. Depending on who you ask, it could be a third or maybe a half of all people walking around right now perfectly healthy. You could actually find C. diff in their gut, but it's usually in very small numbers. It's not a huge amount. But when you take those antibiotics, they wipe out that balance. The microbial antagonism isn't taking place. C. diff now grows to a much higher amount than normal, and because the balance is wiped out, the C. diff now has more room to grow, and because of its specific effects, it causes damage over time, causing its own disease, which was ultimately triggered by the antibiotics, which are supposed to keep bacterial infections from happening in the first place. So it's kind of an ironic set of events that happens there. Last thing here is kind of where does this all start? I said earlier, as an adult, you have this sort of complex ongoing relationship. But the truth is, where a lot of emphasis is being placed, in addition to the gut health, is at the point of birth. And there's some debate about exactly how this works. But generally, it's thought that when you're first born, you're more or less sterile. Whether that's exactly right or not is kind of up for debate. You may have some microbes, as this point mentions here. It could be that you actually have some level of microbiome before you ever are born. But either way, it's generally thought that at the moment you're first born, you have very few, if any, microbes established in the body. And so when you're first born, that this process is called parturition, giving birth. And it's almost like opening up a sterile petri dish to the environment. The dish has been perfectly kept sterile, and now you take the lid off, and all of a sudden microbes can be introduced and colonization starts. And whether you're perfectly sterile or not, either way, you're largely being introduced to the majority of microbes within the first moments, really, of being born. And then colonization takes place heavily within the first weeks or months after you're first born. So some of the emphasis then has now been placed on, well, how are you born? You know, traditionally, you were always delivered through the vaginal canal. Today, you can be born, and many are, by a cesarean section. And kind of like the whole conversation about awareness, for many years, people were having, C-section births, cesarean births, without much attention to it. I mean, the child's born, so who cares? But now with this awareness about the microbiome, and especially about how you're born more or less sterile, they're now recognizing that the way you're born will affect the colonization of the body. If you're born through the vaginal canal, you're going to be exposed to microbes through the vaginal canal in a way that's different than if you're born through a C-section. You're essentially bypassing that entire natural process to be born through C-section. And it turns out that that makes a difference in how the body is colonized. Now, some of the questions about that are, how much does that matter? And that's where you get into ongoing multi-decadal research to really hone in on what the exact differences are. But early research has shown and largely suggested that the difference has an effect on your immune system. So when you're first born, your immune system is still developing. In fact, your immune system really develops pretty heavily up until around the time you're about 5 years old, and really even more up until around puberty. And there's different opinions on that, but really within the first 5 years, your immune system is heavily developing for sure. And so the exposure to microbes within those first so many years turns out to shape the immune system in kind of these interactive ways. And so one of the ways this is thought to have an effect is on quote unquote immune tolerance. The immune cells need to kind of learn what to attack and what to leave alone. They have certain little signals that tell them attack these cells over here, but leave these cells alone. Don't attack these, attack these. That's the idea of immune tolerance. And so it seems as if the presence of microbes very early is shaping that sort of a system of who to attack and who to leave alone in a way that's very complex. And that's another part of the ongoing research. How exactly does that work? That's hard to say. But there seems to be some effect on how the tolerance is shaped depending on the mode of birth. So the vaginal birth seems to expose you to the kind of quote unquote natural microbes. You know, that's the way it's always been done. So your body has sort of adapted to that process. And so that's kind of the normal, correct way of doing it. And if you go through a C-section, now your body is being inoculated with a different set of microbes that are not quote unquote normal. And so the immune system seems to have differences in the way that tolerance is developed, in that the correlation there is that babies born through C-section are more likely to develop things like asthma, obesity, eczema, diabetes, and others. Now, correlation is not necessarily causation, and that's another part of the trickiness here. Is it some other factor, or is it strictly the cesarean section? Because whether you're given breast milk, or whether you're fed infant formula, or whether you have dogs or cats in your house, or whether you have carpet, or whether you have hardwood floors. Believe it or not, all those things make a difference. Whether you play outside and whether you get dirty as a child, or whether you're indoors and clean more often, all those things contribute. What kind of food you eat. So it's never as simple as this one thing causes this outcome. And that's part of the difficulty in making these determinations. But correlation is definitely there. There's a correlation that if you're born through a C-section, you're more likely to have a series of these different types of diseases than if you're born through a vaginal type of birth. So whether that's the sole contributing factor is a different question. And the last thing here is what you can do about that. So because this awareness is becoming more and more prevalent, there are different opinions about what you can do. And these opinions here come from a pediatrician named Dr. Mark Sloan, who's just sort of suggested some ideas that if you're born through a cesarean section, you can potentially use probiotics. You don't have the normal bacteria, so try to bolster that by somewhat artificially by using probiotics. This one's a little more kind of gross and weird, but you can actually transfer maternal secretions. So if you're born through the vaginal tract, you're going to be picking up bacteria through these secretions. And if you're going to avoid that, perhaps do that artificially. Take some of those secretions and artificially introduce them to the child to kind of compensate for what you missed through the cesarean section. And even going more extreme is it was called a fecal transplant. And a lot of people are totally grossed out by this, but the idea is that feces actually has these bacteria in them. And really, feces are kind of like a probiotic. Again, very gross, but the truth is that in the feces there are normal bacteria. Many of them are good. So if you can find a way to try to transfer those back in through this way, it's sort of like a probiotic. And there's just a lot of issues with that. You know, people aren't open-minded to that, obviously, and there's also harm that comes along with that. So, but that's sort of like a probiotic, believe it or not, doing a fecal transplant. So if you're born through cesarean section, these are things that you could attempt to do to kind of reestablish that normal balance back to as if you've been born vaginally. So, in the next part, we'll take a look more at infection and kind of the traditional interaction of the body in terms of germs and disease and things like that. But this lesson here has been kind of about the new age awareness of the microbiome and some of the interesting things that go along with that. So we'll wrap it up here and we'll do part 2 on infection.